Okay, Shane, so I'm going to walk you through a couple of things. So what I typically do from the eScience Labs, you can see up here, I went to eScience Labs, the Lab 4, and then clicked done. Well, I went through each of the links. I think there was 4 of them. This is the third one, Experiment 1, Qualitative Analysis of Reactions, and this is where the instructions are. But then the other thing, of course, which as we and you seem to recognize, is that our instructions take precedent. So as it says here, you start with accessing 5.1, the introduction, just read through that. That just explains some things about the overall concept in theory. And then you go ahead and complete the experiment. You're going to test for hydroxide and hydrogen ions, and 5.2, experiment 1. So that's what I'm showing right here, qualitative analysis of reactions. And right here, part 1, we're going to test for the hydroxide and the hydrogen ions. So this is really where you're going to start the actual chemical experiment. And as it says, use your pipettes to add the drops, and then when you're done, rinse them, blah, 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 blah. Okay, but then down here, so obviously I know you don't need me to read through this, but your question was about adding the acid. Use a pipette to add 10 drops of the acetic acid to the test tube labeled acetic acid. Okay, so you're going to be using your pipette to add the acid. And again, I would just go back and forth kind of between the experiment instructions, the APUS ones that are right here. There's some tips and things like that. And then over here, what you're doing. Okay, so the overall idea is you're going to be testing known solutions for things, hydroxide and we call these protons or hydrogen ions. And then you're going to test the knowns for carbonate ions being present, and you'll see what it looks like when the particular solutions have these ions, these different ions. Then you do the sulfate, then you do the phosphate, then you do calcium, and then you're basically going to do the same thing with an unknown, which is something you don't know what it has in it. So you use these initial ones. When you know calcium is present, you follow the steps and say, okay, this is what it looks like when calcium ion is present in a solution. I know this solution has calcium. Here's what it's going to look like when I do these things to it. And then when you come down here and do the same things to your unknown, when you do the same steps and you get to this part right here where you're adding the calcium acetate, doing all that, or sodium fluoride, excuse me, well, that's what you're doing up here. But when you do this part and you match it, does it do the same thing? Does your unknown do the same thing that your known did? If yes, it must have calcium. If not, then it doesn't have calcium. So that's kind of the overall what you're going to do. Now, there are some tips and hints. I'm going to come back here real quick on the APUS instructions. Again, use the pipette to add the drops, and when you're done, you're going to rinse the test tube with distilled water, not tap water. It talks about making sure your test tubes are clean and dry before you do anything else, and that's because you could get false results. Even our tap water has stuff in it, has ions. Distilled water doesn't. Note 2, it says rather than dipping your litmus paper into solution and unnecessarily soaking it completely, use your pipette. Just like stick your pipette in there, the tip of it, and then drop it on, put a drop or 2 onto the litmus paper. That's it. And then you're going to take a picture of you holding your results, meaning the reacted litmus paper, and then your label, if you can do that, that selfie kind of thing, and then that'll go in class. Then you're going to do the qualitative analysis of reactions test 2 for hydroxide and protons. And again, basically the same kind of warnings or notes here. Then you're going to do carbonate. Then you're going to do sulfate, phosphate, calcium. So see, they do kind of match up with what's here. All right, what's here? Test for hydroxide and protons, test for carbonate, test for sulfate, test for phosphate, test for calcium. And then we're going to do the second part, determining of an unknown. And again, you're going to see the same notes. Again, make sure you're always using distilled water, clean your test tubes really well, make sure you use clean, dry pipettes as well. You don't want to contaminate anything like that. Okay? And then pictures at each step along the way. I'm hoping that helps. And then as it says, make a summary table. You can actually come down to the bottom of this page, download the tables that you need, and again, it's going to match with, you know, certain questions are going to ask for certain tables, so just make sure you're doing the right ones. And then again, you do the post-lab questions just in preparation for what's going to be asked of you. At the end, you should have 8 PDF documents, one for each of the 6 tests that you do, one for your unknowns, and one with your summary table, okay? And in fact, you know what? I take that back. Instead of downloading all these separately, this is the summary table. My apologies, I spoke wrong. Just print this out, fill it in, or, you know, use an Adobe editor and fill in the boxes. Either way, you can print it, fill it out by hand with a pen, and then take a picture, or you can, you know, use Adobe Editor and fill in the boxes. Whatever you want to do. But this is your summary table. So that's all you've got to do. You're going to be submitting a bunch of pictures and submitting this summary table. All right. I hope that helps. Let me know.